Hi students, welcome to the first part of the Chapter 10 lecture. Today we're going to be covering the structure and function of DNA. So what exactly is DNA? Well, DNA stands for Deoxyribonucleic Acid, and it's the molecular basis for the genetic code of all living organisms. So regardless of whether that organism is a bacterial cell, a plant, an animal, a protist, a human, all of those organisms share this molecular basis, and that is DNA. It was known as a chemical in cells by the late 1800s. It has the capacity to store genetic information. It's copied and passed from generation to generation. And again, the genetic code is shared by all organisms. So everything that's alive on this planet that has all of those 7 properties of life that I introduced you to during the first lecture has DNA. And a genome refers to the entire sequence of an organism's DNA. DNA as well as RNA are nucleic acids. That is one of those 4 categories of macromolecules that I introduced you to a few lectures ago. They consist of chemical units called nucleotides, and nucleotides themselves consist of a phosphate group, a nitrogenous base, which is either A, T, C, or G, and a sugar. The sugar-phosphate backbone links nucleotides together. So you should be familiar with this a little bit because I showed a similar schematic to you in another lecture. But the sugar here is blue, the phosphate group is yellow, and then there's one of 4 nitrogenous bases. So this would be a single strand of DNA within a double helix. So we have the phosphate group, a nitrogenous base, and a sugar. And again, those nitrogenous bases are adenine, guanine, cytosine, or thymine in the case of DNA. And in the case of RNA, uracil takes the place of thymine. 2 of these are double-ringed structures, adenine and guanine, and 2 of them are single-ring structures, cytosine and thymine. So the 4 nucleotides that are found in DNA differ in their nitrogenous bases, and I already went over that, but that's how you spell all of those different nitrogenous bases. And RNA, which is ribonucleic acid, has uracil in place of thymine. James Watson and Francis Crick determined that DNA is a double helix, and this was back in the 1950s. And Rosalind Franklin collected the X-ray crystallography data, which actually produced images of DNA. The model of DNA can be likened to a rope ladder that's twisted into a spiral. So we have the 2 strands here made up of that sugar-phosphate backbone, and the rungs of the ladder would be the nitrogenous bases, and those nitrogenous bases pair with one another and hook up in the middle. Again, notice that the bases pair in complementary fashion. Adenine always pairs with thymine, and cytosine always pairs with guanine. So we have a double-ring structure pairing with a single-ring structure. So what exactly is DNA's purpose? Well, it's simply to make copies of itself. When a cell or organism reproduces, a complete set of genetic instructions must pass from one generation to the next. So regardless of whether an organism reproduces asexually or sexually, there has to be a complete set of genetic instructions inherited by that next generation. So if it's an asexual organism, it simply duplicates all of its own DNA, and the offspring is basically a clone of the parent. With sexual reproduction, there's some recombination, of course, as I've talked about rather extensively with the meiotic process. Alleles will be passed down in sperm and egg cells, and then those alleles combine with those that are in the complementary gamete. So with sexual reproduction, there's a lot more variation that's possible. Replication is the process of DNA producing two daughter strands from one original parent strand. So here we see the parent DNA molecule shown in blue. What happens is there are special enzymes that will split that DNA molecule in half without damaging it and allow replication to occur. So replication occurs with the nitrogenous base here. Of course, if it's a G, the new base that has to pair with that would be a C. If it's an A here, it has to be a T. So the new daughter strands are added to that single strand from the entire genome from the parent molecule. So DNA replication begins at specific sites on a double helix. It's not just at the end. So we can have several origins of replication occurring. Again, the parental strand is shown in blue and the daughter strands are shown in orange. So what you end up with is two daughter DNA molecules. One half is composed of the original parent strand and one half is composed of the newly synthesized daughter strand. And the replication process can actually proceed in both directions. So it can initiate replication in the middle here, and rather than just going one way along the original DNA strand here, it can actually proceed in both directions simultaneously. DNA functions as the inherited directions for a cell or organism. But how exactly are these directions carried out? So if DNA is the blueprint, how does that blueprint actually get built? Remember that an organism's genotype is its genetic makeup, and this is based simply on the sequence of nucleotide bases in DNA. The phenotype is the organism's specific traits that are expressed, and by expressed, I mean that there's some physical manifestation of those traits. Whether it be blue eyes or curly hair or freckles or a certain amount of insulin produced, all sorts of things. So it's not necessarily just physical characteristics that you can see, but it's any genetic code that is actually expressed within the body. So it could be invisible to us, but it does have some physical manifestation. DNA specifies the synthesis of proteins in two different stages, transcription and translation. And by synthesis of proteins, this is basically just to say that for any trait to be expressed, there has to be proteins made from that genetic basis. So remember when I was telling you all about proteins are not just muscle tissue or something like that that's necessarily obvious. It's all sorts of specific structures within our bodies and within all living organisms. So proteins basically are really important building blocks for all of life. So in order for the instructions within DNA to be carried out, you have to actually have some sort of process that takes that information and translates it into a protein. And these two processes are responsible for that. So we have transcription, which takes place within the nucleus, in which an RNA molecule is made from a DNA template molecule. And then we have the process of translation, which takes place in the cytoplasm outside of the nucleus. So we have that RNA template that has basically transcribed the information within DNA, and in turn that RNA molecule is translated into a functional protein. Transcription is the transfer of genetic information from DNA into an RNA molecule. And this will take place, again, it's within the nucleus. We have the parent strand in blue here, and we have the newly made RNA in red. And remember that RNA has uracil instead of thymine. So when the nitrogenous base on the parent DNA strand is adenine and it's undergoing transcription, you're actually going to have uracil pairing with that adenine rather than thymine. This takes place within a special enzyme, which is also a type of protein, known as RNA polymerase. And again, all of these little individual pieces here are RNA nucleotides that are being added to the newly made RNA strand. So in transcription, genetic information is transferred from DNA to RNA. You can think of RNA as the middleman between DNA and actual proteins. An RNA molecule is transcribed from a DNA template. DNA does not become RNA. There's actually a brand new strand of RNA made based on the DNA blueprint. Okay, so the second step of this process is known as translation. And entire biology book chapters are devoted to these processes of transcription and translation. You could write an entire book about the process just because it is so complicated and there's so many different molecules involved. But for the purposes of this introductory class, I'm not going to go into too much detail. So basically, what you need to know is within these slides here, and pay particular attention to the bolded words. Okay, so translation is the transfer of the information in RNA into a protein. So again, RNA is like that middleman. It has transcribed the information from DNA, and that information is now stored within RNA. So we need to get from RNA into a functional protein. So the information or language within DNA is ultimately translated into the language of polypeptides, which are the building blocks of proteins. And a codon is a triplet of base pairs that code for specific amino acids. So three nitrogenous bases here make up a codon. In turn, a codon codes for one particular amino acid. So this process occurs in groups of 3. And again, this is the overall schematic of this transcription and translation process. A DNA strand acts as the template for transcription. An RNA strand is made from that template of DNA, and then that RNA strand is translated into a polypeptide, which is the building block of a protein. Alright, I'm going to go ahead and pause here and pick up in the next section.